network, charcoal, resin, tar, and potash. Acronym of our action is EU-PoTaRCh, please remember. And the number of our action is CA22155. This is full name of our action. The reason for today's meeting is short-term scientific mission in which Marina Chavenetti-Dow participated. Let me introduce in short Marina. Marina is from the Laboratory of Forest Utilization, Department of Forestry and Natural Environment, Aristotle, University of Thessaloniki in Greece, of course. Marina, did I say everything correctly?
Yes.
Thank you very much. Thank you. Marina spent almost 2 weeks in Germany at the Dendrochronological Laboratory in Stuttgart. Host of this term short scientific mission was leader of working group number 3, Archaeology, Oliver Nelle. During this stay, Marina, of course, work with our deliverables, goals of EU-PoTaRCh Action, because this is obligation. All activities we have to organize in collaboration with connection with our deliverables, which you can find in Memorandum of Understanding, and of course on our website. And I hope that Marina will tell you more about details. And the technical details, so this meeting is recorded, I think. Kuba, it's recorded?
Yes,
probably
it is.
yes.
I
Okay, thank you.
Started.
And after this, after presentation of Marina, you will have possibility to ask questions. So we have few minutes for questions, deliverables, comments, etc. So I think that we can start. Marina, the floor is yours.
Thank you. Good morning. My name is Marina Chavenetti-Dow. As Dr. Zborowska said, I'm a laboratory teaching staff at the Aristotle University of Thessaloniki. My STSM was conducted at the Technological Laboratory of Hemmenhofen under the supervision of Dr. Oliver Nelle. The issue was on charcoal analysis from archaeological regions of Germany. Since my specialization is on wood anatomy, when I wanted to apply for this as a cost action, I was looking for an institution that was working on charcoal analysis, the chronological research, and relative with wood. So I tried to find among the management committee the institutions or universities that were working on relative researches. I contacted Dr. Nelle after checking his research work and publications, and saw that we had some similar research interests. And I contacted him and asking him whether we could collaborate and whether he would accept me to work with him in a relevant topic. After he accepted me, I formed a proposal, a research proposal related to the research objects, research interest of the institute. And I applied for this STSM. The research took place at the Technological Laboratory of Hemmenhofen. The map shows where the laboratory is located, nearby the Lake Constance and close to the Switzerland borders.
Sorry, Marina, could you show us next slide?
Yes, I did. I am at the next slide.
Because I see first one.
Yes.
Okay, now it works. Okay, thank you very much. Now we can see map.
Now you can see the map. Should I turn it to the presentation? Do you see it now?
I think there is a problem if you share the screen and you are not in the full screen mode of the presentation, then it will show only the slide on which you start. Unless you open the presentation in a full screen and then share like this screen.
It's full screen and full screen at my presentation. Can you see it now?
We see now the PowerPoint.
Okay. Give me a minute. Yes. So give me a minute to do it again then. Sorry for the delay.
No problem. With the Teams, it's usually the problem with the sharing of the full screen of the presentation.
When I use this window and I do the presentation profile, it doesn't give me the chance to have any other changes. Do you see it now? Can you see the presentation?
Yes, third slide.
Third slide. Do you see me when I change the slides?
Yes, but yes, so yes, you have to change the slides. And so I will tell you if we will see, okay, first slide,
second slide,
second
here.
slide, and
I'll do it this way.
Okay.
Yes, maybe. Yeah.
We can work in this way. Perfect.
Yes, we can. So this is the map where it shows where the laboratory is located, as I said, near Lake Constance and close to the borders of Switzerland. The aim of this STSM was the study of charcoal from late medieval and early modern times in order to identify the species of the trees which were used for the production of charcoal in the past, so as to obtain information about the exploitation of the vegetation through time. Furthermore, the study of charcoal gives information related to climatological conditions of the past by studying trees' growth through information. So the research included taxonomic charcoal analysis and tree measurements with the aim to identify the wood taxa used in these regions. As a starting procedure, in order to learn how to handle charcoals and work with them and get an overview of the wood anatomical variation, samples from the excavation Isnang under Eichen at Lake Constance were studied and identified with the use of stereoscope and reflectance light microscope equipment. The first picture shows the stereoscope that was used for the identification of the samples. As we see, we have a sample of fragment of charcoal under study. The next picture, the second one, is the microscope we used, and the third shows the whole equipment, how it's connected to each other. A camera attached to either the stereoscope or the microscope gives the opportunity to capture the image through a screen which is connected with a computer. So in each case, we can capture images either microscopic or macroscopic at stereoscope. The fourth image shows charcoals from previous findings, which are preserved for study. In order to study the charcoals, we start with a knife, and we use it to produce freshly broken surfaces in order to have the chance to observe them in reflectance light with a stereo lens and the microscope. After the identification, the samples are preserved in paper envelopes, and as we saw, the identified species included the species Fagus, Quercus, Corylus, Salix, Fraxinus, Alinus, and Abies among them. The first picture shows the procedure with a knife, how we produce smooth surfaces on the charcoal. The second on the top and the third one at the bottom show images of Fagus and Quercus through the magnification lens of the stereoscope. And the fourth is the envelopes at which the samples are preserved after the identification. This is an image of Salix willow at the stereoscope. we can see all the anatomical features of wood. Here we have diffuse pores, and the vessels, the main cells of broad-leaved trees are apparent. And since they are since it's diffuse pores, they are evenly distributed all over the surface. Among the vessels are smaller cells, which are the fibers, and we can see easily the rays here, these lines which consist of parenchymatic cells. At this slide, we can see microscopic pictures of alnus, alder, aesculus, horse chestnut, and carpinus, hornbeam, at different magnifications. The difference between charcoal and wood is that in the case of charcoal, we can produce thin slides in order to see all the anatomical features of wood. We only produce smooth areas that we can check some of the characteristics of the wood. In the second step, at fieldwork, charcoal samples were collected from the Black Forest South Freiburg in the Sonsland region during a field trip to the area. After digging the soil, samples were collected at different depth profiles, including surface samples, so as to identify the species and check the evolution of their exploitation through different periods of time. The first image shows a view of the Black Forest, and the second shows the depth of the hole that we dug. After the collection of the samples, they were preserved they were collected in plastic bags, and then were left to dry in open boxes. It was sunny these days, until their humidity reached the levels of relative environmental humidity, and was easier to handle them without collapsing. Afterwards, the same procedure as before was followed, which means that we use the stereoscope and microscope for the identification of the samples. And the findings suggested that the main species used for charcoal at this period, at this area, were Fagus and Abies, Beech and Fir. Moreover, measurements of tree ring width were conducted with tree ring measuring tables, such as the one that is shown at the picture, so as to correlate the width with the predominant climatological conditions of the area in the past. The pictures that follow are from the samples collected at the field trip. This is Fagus from the area. It's easy to identify Fagus since it has diffuse pores with very predominant arrays, which consist of many series of parenchymatic cells, and its diffuse pores with clear ring widths. And the second species which was obvious, which was found at the field trip collected from the samples, was Abies. Again, in this case, we didn't have the chance to have large surfaces to collect, and we have to identify from smaller surfaces. So the main achievements of my STSM, our STSM with Dr. Nelle, the collaboration resulted in gaining of new knowledge for me in relation with the methodological development of charcoal analysis and identification. The practical training carried out in the laboratory focused on a good anatomy and taxonomic identification. We exchanged scientific views and experience and had the opportunity to establish a network for further communication. And we have already talked about with Dr. Nelle that the data we collected during a CSM can lead to scientific publications, especially to publish a paper on the detailed analysis of the samples from K600 from the Black Forest of Sonsland region. Thank you for your attention, and in the end, I want to thank very much Dr. Nelle for our collaboration. It was a pleasure working with you, Dr. Nelle, and I enjoyed my work there. Thank you very much.
Thank you very much, Marina, for presentation. I see that you spent very intensive and interesting time in Germany, so in Stuttgart, in forests. So I see that you performed a lot of different investigations and you detect a lot of different species. So
It was
I
very
believe
interesting. Yes.
I believe that this time was for you very, very valuable. And I hope that you maybe will collaborate longer with Oliver and you will continue with some publication maybe or something like this. So it will be very, very good effect of our cost action also. So I hope that you will use these results and this also this time your experience also in the future. Okay, so now we have few minutes for questions, for comments, for doubts. So are there any questions for Marina?
Yes, please, Magda. It's Oliver.
Oliver, nice to see
hello.
you. Oh,
Hello,
nice to see you.
Thank you, Marina, for the stay was great. Your work is great, and the presentation I also very much enjoyed. And
Thank you very much.
I think it was a very productive time you spent with us, and we are in the process of having a publication on the specific work you did on these charcoal production site charcoals. Our collaboration will continue, and we also talked about that in 2026,
yes,
there will be the International Anthropology meeting, which is every 3 to 4 years. The community of charcoal and analysis specialists will gather in Thessaloniki. So we were also starting to think about having maybe some cost action site meeting of this international
will be great
meeting, which would very much fit together because of the subject and yeah. So thank you, Marina, from my side.
Thank you very much, Oliver, as well. I will be glad to help you organize a meeting here at 2026, and we will be working, we'll continue our collaboration. It was a very nice experience for me, and I collected valuable knowledge. So I would like to continue working with you, and it's a great honor. Thank you.
Okay.
It's nice to see you.
I see big networking potential in this collaboration. So I'm really lucky because this is very, very important part of our action networking. And I see that you are able to collaborate and create network between your institutions. So this is for me very, very, very good information. Thank you very much. And do you have are there any
Yes.
other questions? Yes.
Can you hear me?
Yes.
Yes. Hello. Thank you for your presentation. I'm Eric Sansonetti from Latvia. I just wanted to ask you if there were any critical moments or problems. Maybe could you make all your job according to plans, or there were some problems or something unexpected during this mission?
No, actually, we didn't have a problem. We were very well organized. We had communicated with Oliver since before I got in Germany. So we had a plan before I get there,
Yes.
and everything went according to the plan. We
Mm.
did the field trip, we collected samples. The previously of the field trip, I had the chance and the time to study charcoal so as to get acquainted with it, and it was easier to handle charcoal at the field and the collected samples afterwards. I think we didn't have any problems, and everything went very well. I think Oliver
Really?
agrees with me.
Okay,
Yes.
thank you.
And
Really
we
Thank
good
you.
had
cost. Really good
measurement
cost.
test. Yes, everything was perfect. Yes.
Okay.
And he had very good colleagues who helped me as well, and we had a nice collaboration very well. I worked
Okay.
very well at the laboratory. Yes.
Thank you.
So
Thank
this
you as
is
well.
second STSMs hosted by Oliver. So Oliver has big experience, yes, and I see that this collaboration is really good with Oliver and with institution generally, if of with Oliver's institution. Yes, so it's really good. Okay, other questions, comments? If not, just a reminder that all information on how to organize STSMs and how apply you can find on the EU-PoTaRCh website, of course. And of course, you can ask us for everything. And Katia Tika, our grant awarding coordinator. So if you need more information how to organize STSMs, please contact us and we will support you for sure. So please try, please apply your application, and in the range of core group we will discuss your application and we will send you our decision. So please try, please apply, and I'm sure that this kind of activity is very, very important and very valuable for you. Thank you very much, Marina, for
Thank you
your
very much as well.
very good presentation. Thank you, Oliver, again for your support, and see you during next webinar or training school or other activity which we organize in the range of our cost action. Thank you very much. Have a good day.
Thank
You
you.
too.
Thank you.
Have a nice
Bye
day.
bye.
Bye.
Bye bye. See you.
Thank you.
Thank
Bye.
you. Goodbye.
Thank you. Bye. Kuba, thank you very much for support.
Thank you. Goodbye.
We're waiting for you in Thessaloniki, and I hope I'll have the chance to visit you in March.
We are
So
waiting.
we have the chance.
We arrange something. We will
Yes,
arrange something.
yes. Okay, nice to see you. Have a nice day.
Thank you. Bye.
Bye.
Bye.